The process of chromosome alignment during cell division is one of the most meticulously choreographed events in biology. At the heart of this process lies the centromere, a specialized chromosomal region, and the kinetochore, a proteinaceous structure that serves as the interface between chromosomes and the mitotic spindle. These components work together in a highly dynamic and regulated manner to ensure the accurate segregation of genetic material. The precision of this process is crucial for cellular viability, as errors in chromosome alignment can lead to aneuploidy, a hallmark of many cancers and developmental disorders. The importance of precision in chromosome alignment cannot be overstated. Each step in this process is critical for maintaining the integrity of the cell's genetic information. Any deviation from the norm can result in severe consequences, including cell death or the development of diseases. Therefore, understanding the mechanisms that ensure such precision is vital for both basic biology and medical research. Understanding the molecular mechanisms governing centromere and kinetochore protein dynamics during chromosome alignment has been a subject of intense research. High-resolution imaging techniques, live cell microscopy, and proteomic analyses have provided crucial insights into the spatio-temporal coordination of these components. This documentary explores the intricate molecular interplay at the centromere and kinetochore, shedding light on the fundamental principles that govern chromosome alignment and segregation. The centromere is an essential chromosomal locus that serves as the site for kinetochore assembly. Unlike other chromosomal regions, centromeres are epigenetically defined rather than determined by DNA sequence alone. The presence of the histone H3 variant, centromere protein A, CENP-A, is the primary determinant of centromere identity. Studies using chromatin immunoprecipitation sequencing, ChIP-seq, have demonstrated that CENP-A nucleosomes are enriched at centromeric chromatin, providing a stable platform for kinetochore formation. The structural organization of centromeres varies across species. In humans and most eukaryotes, centromeres are composed of long stretches of repetitive alpha-satellite DNA, whereas yeast centromeres are defined by a single nucleosome-sized sequence known as the point centromere. Despite these differences, the fundamental role of the centromere in chromosome alignment remains conserved. Experimental depletion of CENP-A results in the failure of kinetochore assembly and subsequent chromosome missegregation, highlighting its indispensable function. Beyond CENP-A, centromeric chromatin is enriched with other centromere-associated proteins, such as CENP-B and CENP-C. CENP-B binds to alpha-satellite DNA and facilitates the recruitment of additional centromeric proteins, while CENP-C acts as a scaffold linking centromeric chromatin to the inner kinetochore. Fluorescence recovery after photobleaching or FRAP studies have shown that CENP-C exhibits relatively stable binding at centromeres, reinforcing the structural integrity required for kinetochore assembly. The kinetochore is a multiprotein complex that assembles at centromeres and mediates the interaction between chromosomes and spindle microtubules. It consists of an inner kinetochore, which is directly associated with centromeric chromatin, and an outer kinetochore, which captures and stabilizes microtubule attachments. The assembly of the kinetochore occurs in a stepwise manner, beginning with the recruitment of the constitutive centromere-associated network, or CCN, a group of 16 proteins that act as the foundation for kinetochore formation. A crucial component of the outer kinetochore is the KNL1-mis12NC80, or KMN network, which directly binds spindle microtubules. The NDC80 complex, in particular, is the primary microtubule binding unit and plays a key role in maintaining stable kinetochore-microtubule attachments. Live cell imaging studies have revealed that NDC80 undergoes dynamic phosphorylation by Aurora B kinase, a key mitotic regulator. This phosphorylation event weakens microtubule attachments, promoting error correction and ensuring proper chromosome alignment. The kinetochore must establish end-on attachments to spindle microtubules, a process that is regulated by multiple microtubule-associated proteins. Dynin, a -N-directed motor protein, facilitates initial lateral kinetochore-microtubule interactions, while the microtubule plus N tracking protein EB1 stabilizes mature attachments. Defects in these mechanisms lead to lagging chromosomes and chromosome misalignment, which can have deleterious consequences for genomic integrity. One of the most fascinating aspects of chromosome alignment is the role of mechanical tension in stabilizing kinetochore-microtubule interactions. Proper bioorientation, in which sister chromatids attach to microtubules emanating from opposite spindle poles, generates tension across kinetochores. This tension is sensed by the spindle assembly checkpoint, or SAC, a surveillance mechanism that prevents premature anaphase onset. Aurora B kinase plays a pivotal role in this process by phosphorylating kinetochore proteins in response to low tension. Phosphorylation of NDC80, for example, weakens microtubule attachments, allowing error correction to take place. When proper bioorientation is achieved, tension pulls kinetochores apart, reducing Aurora B activity and stabilizing microtubule attachments. This dynamic phosphorylation-dephosphorylation cycle ensures the fidelity of chromosome alignment. Recent studies using Forster resonance energy transfer, or FRET-based biosensors, have provided direct evidence of tension-dependent Aurora B activity. These experiments revealed that Aurora B activity is highest at kinetochores experiencing low tension and decreases as stable attachments are formed. This feedback mechanism is crucial for preventing chromosome missegregation, as loss of Aurora B function leads to persistent kinetochore-microtubule attachment errors. The spindle assembly checkpoint, or SAC, is a critical regulatory pathway that monitors kinetochore-microtubule attachments and prevents anaphase onset until all chromosomes achieve proper alignment. SAC activation occurs when unattached or misaligned kinetochores recruit checkpoint proteins, such as MAD1, MAD2, and BUBAR1. These proteins inhibit the anaphase-promoting complex, cyclosome, also known as APCC, delaying mitotic progression until errors are corrected. Live cell imaging studies have shown that SAC proteins rapidly localize to kinetochores lacking stable microtubule attachments. Once all kinetochores achieve proper alignment, SAC components are removed, allowing the cell to proceed to anaphase. Mutations in SAC proteins have been implicated in chromosomal instability and tumorigenesis, emphasizing the importance of this pathway in maintaining genomic integrity. One of the most striking examples of SAC dysfunction is seen in cancer cells, where chromosomal instability arises due to aberrant SAC activity. Studies using cancer cell lines have demonstrated that overexpression of MAD2, for instance, leads to prolonged mitotic arrest and aneuploidy. Conversely, depletion of SAC components results in premature anaphase onset and chromosome missegregation, further highlighting the delicate balance required for accurate chromosome alignment. The study of centromere and kinetochore dynamics has been revolutionized by advances in high-resolution imaging and proteomic approaches. Super-resolution microscopy, such as structured illumination microscopy and stimulated emission depletion microscopy, has enabled researchers to visualize the nanoscale organization of kinetochore components in unprecedented detail. Additionally, quantitative proteomics has provided insights into the composition and post-translational modifications of kinetochore proteins. Mass spectrometry-based studies have identified phosphorylation, acetylation, and ubiquitination events that regulate kinetochore function. These findings have expanded our understanding of the molecular mechanisms governing chromosome alignment and segregation. The dynamic interplay between centromere and kinetochore proteins ensures the faithful alignment and segregation of chromosomes during cell division. From the epigenetic regulation of centromere identity to the tension-dependent stabilization of kinetochore-microtubule attachments, every step of this process is tightly coordinated. Disruptions in these mechanisms can have profound consequences, leading to chromosomal instability and disease. As our understanding of centromere and kinetochore dynamics continues to evolve, new therapeutic strategies may emerge for targeting mitotic defects in cancer and other genetic disorders. By unraveling the molecular intricacies of chromosome alignment, researchers are paving the way for novel interventions that safeguard genomic integrity and cellular homeostasis. The story of centromere and kinetochore proteins is far from complete, but each discovery brings us closer to deciphering the fundamental principles of cell division.